Okay, it is 1:05, so I'm gonna go ahead and get started. Excuse me if I stutter, but that's probably just me taking a pause to let somebody gain access to our meeting room. But welcome our citizen scientists to our first virtual lab experience. And my name is Dr. Candace Johnson. You can call me Dr. Candace, that's fine. And I routinely work at the South Hill site. And we also have a site in the city, which Miss Carrie Ann and Miss Cynthia teach over there. So they're in the chat and they're also logged in. So feel free to speak with them or go ahead and say hello. But today's activity is going to be Twizzler DNA. So yeah, Twizzlers are really yummy, and DNA is a very important molecule in our lives. It helps keep us alive, and it makes us who we are. So today it's just going to be an introduction to DNA and also DNA structure. And we're going to learn a bit about that by building our Twizzler DNA model. So just a little bit of housekeeping rules. Again, if you have a question, please use the hand raising feature or send me a message or send the group a message, or just as a parent did, try not to interrupt, but if it is really pressing and you have a question, you can go ahead and unmute yourself and go ahead and speak. But just to keep it nice and streamlined, I would like for you to use the messaging feature for all of your questions and comments, and try to keep yourself on mute so that your background noise does not interfere with everybody else's activity. In your registration email, you should have received a list of items that you would need to participate in this lesson. If you do not have those items, that is okay and fine. You could just watch the videos and ask any questions that you might have about the structure of DNA. So we're gonna get into the materials in a second, but first things first, I want to know how much you guys know about DNA. So in just a second, I'm going to release a poll. So you should see a window pop up on your screen. And this is kind of just a pre-test to how much information you know about DNA in general. All right, so first things first, we're going to do a Zoom poll, and I'm going to see how much information you guys know about DNA. And then after today's experiment, we're going to take another poll just to make sure that we learn the basics of DNA, okay? So I have launched the poll, and the poll has 5 multiple choice questions, and you are to select the answer that you think is correct. And Becca's family, I guess you guys can work as a team over there. But select the answer that you think is correct. You guys choosing answers out there? I'll give you about 5 minutes to participate in the Zoom poll. Okay, about half of you have gotten through your polls. You have about 3 more minutes for questions to be answered. All right, about 1 more minute. See if you can get your answers in. All right, couple more seconds. All right, one more person has to finish up and then we will move along. And just so you know, I won't be sharing the answers with you right now. So these are questions that if you don't already know, you're going to learn about within our class. So we're going to take another poll at the end of our activity. And this should test and see if you guys picked up any information while we were busy having fun. But looking at the results, you guys know a lot about DNA already. There's some things we have to clarify, but you guys know a lot about DNA already. Okie dokie. All right, so I'm going to go ahead and end our poll now. And after our poll, we are going to now get into some background information about the DNA molecule, just so we know exactly how to build our Twizzler DNA molecule. So, again, today we are making a Twizzler DNA molecule, and this is our first week of the Citizen Science Lab's online workshops. So first things first, if you had a chance to gather materials, this activity is really simple. Good job, Becca's family. This activity is fairly simple. We need basically 3 materials. So you're gonna need Twizzlers. And again, if you have shorter Twizzlers, see if you have enough to line up together end on end, and just make sure you have 2 sets of those to represent your 2 sides of your DNA backbone. You're also going to need a bag of colored marshmallows, and if you haven't separated your marshmallows by colors now, go ahead and start separating your marshmallows by color, because it'll make your activity a lot easier when we get into it. Okay, and after that you will also need toothpicks. So very simple. We need Twizzlers, colored marshmallows, at least 4 different colors, and toothpicks. And if your marshmallow colors do not match the colors that I have, that's okay. Just remember which colors you're using to swap in and out. So again, some lab guidelines are to refrain from commenting without raising your hand or sending me a question via messaging. And also try your best to participate in the Zoom polls, which are at the beginning, which we just finished, and at the end of our experiment, and make sure you have fun. And if at any point you're not having fun or you have any concerns, please chime in and make a comment, or you can send me a message via the message box. So very simply, what is DNA? So DNA stands for deoxyribonucleic acid. Now, deoxyribonucleic acid is a material found in the nucleus of all living cells. So inside of a cell, we have organelles. And one of those organelles is called the nucleus. And the nucleus is where all of your genetic material or DNA is stored in the cell. So your DNA is in the nucleus, which is a part of a full cell. So your body is made up of millions of cells. And again, those cells all have nuclei. And in those nuclei, we have our chromosomes. And your chromosomes are your genetic material. And that genetic material is your DNA. And sections of that genetic material are your genes, which make up who you are, how you look, and what you sound like, what you feel like, and also what you like to do. So again, the structure of DNA is very important. So DNA has a specific structure, and this structure allows it to wind up and compact on itself so that all of this information can fit in a tiny, tiny place called your nucleus in your cell. So DNA is basically a sugar phosphate backbone and also nucleotide bases. So your sugar phosphate backbone is made out of a sugar called deoxyribose. You see how that's a part of the name DNA? So deoxyribose is a type of sugar. Now, next to the deoxyribose, you have phosphates, or those P groups with all those oxygens around. And then you have another deoxyribose sugar. So these keep repeating over and over again, and this molecule forms the backbone, or the sides of the ladders of your DNA. Now, the rungs in the ladder, or everything in between, are made up of your nucleotide bases. And in DNA, there are 4 nucleotide bases. We have adenine, thymine, guanine, and also cytosine. So these bases, as you could see, look very different, but they come together via hydrogen bonding to form the rungs in the ladder of your DNA backbone. So with the rungs in your ladder, we always have A's binding to T's through hydrogen bonding, G's binding to C's through hydrogen bonding as well. So they always match up in those specific pairs, and that's what holds your 2 strands of DNA together to form that ladder formation. Now, again, very important, here in this diagram, you see that your backbone have that 5 or that pentagon-shaped sugar, ribose, and also those circles for the phosphates. But in between, you see your nucleotides that are pairing up. So again, your C's always match with your G's, or cytosines with guanines, and that happens through 3 hydrogen bonds seen in those red hash lines. And then your T's always bind with your A's, again seen through the 2 red hash lines through their hydrogen bonds. And again, those hydrogen bonds are what keep your 2 strands of DNA together to form that ladder structure. Now, I'm emphasizing this for a reason, but again, DNA has 4 bases, A, T, C, and G, and these bases have to link up specifically to form a proper strain of DNA. And A's and T's are always buddies, and C's and G's are also always buddies. So, now that we've gotten all of our background information, I'm going to stop and check and see if we received any questions, and then we are going to move into the actual activity. So Questions. Can you use up cut up Sour Patch Kids? I would say yes, you can, as long as you take out four different colors of your Sour Patch Kids, and then you designate which color is which base, okay? So if you're using anything other than the colored marshmallows, yes, you could use any other candy or edible treat, as long as you have four different colors to represent your four different nucleotide bases. So adenine, guanine, cytosine, and thymine. So what would happen if you mixed a C and a T? So, the cytosines and thymines, they would not be able to bond together. So remember, we went over those hydrogen bonds in between our base pairs. So the G's and the C's only come together because they can make those three specific hydrogen bonds. So if you mixed a C and a T, or a cytosine with the thymine, it wouldn't make the proper number of significant hydrogen bonds. So you would have what we call a mispairing in your DNA sequence, which would cause for a little bump in your DNA where the backbone is not held together properly by those inside hydrogen bonds. All right, that's good for the questions. So now we're going to get into the activity. What I have to introduce and walk you guys through this activity is a video. Again, feel free to ask any questions that you need during the video. I'm going to try to stop and pause and allow you guys time to actually create your Twizzler DNAs. But now I'm going to take a second and share this video with you. And it's going to begin again with the materials that you will need. Okie dokie. Now it is time to have your DNA and eat it too. When isolated from a cell and stretched out, DNA looks like a twisted ladder. This shape is also called a double helix. The sides of the ladder are called the backbone, and the steps, also called rungs of the ladder, are pairs of small chemicals called bases. There are four types of chemical bases in DNA: adenine, or C, guanine, or G, and thymine, or T. They form pairs in very specific ways. Adenine always pairs with thymine, A and T's always pair, and cytosine always pairs with guanines, so C's and G's always pair as well. Your task is to use the following materials and procedure to construct an edible model of DNA. Let's go over what you will need. All right, let's get started. First things first, you're going to want to wash your hands, make sure they're clean, because we are creating something that you will probably be eating. So outside of washing your hands, you're going to want to make sure you're working on a clean, dry surface. So now that we have a clean hands and clean surface, first thing you're going to need is 2 pieces of licorice or Twizzlers. Make sure they're nice and flexible for you. It'll help later when you're building. Next thing you're going to need are your marshmallows. So you're going to want to buy a bag of colored mini marshmallows, just like this one seen here. And then what I did, and what helps me a lot, is then to separate that large bag into the 4 individual colors seen here. And that will help with our building. And then last but not least, you're going to need toothpicks. So you're going to need about 12 toothpicks as well. So let's get those ready. And you're going to need about 10 or so of each color of marshmallow. All right, I'll pause here to give you guys a second to either gather your materials or take about a minute or so to separate out your candy or marshmallow colors. So just try to get about a pile of 10 or so of each different color marshmallow or Sour Patch Kid or jelly beans or whatever, or gummy bears, whatever you may be using. So I'll give you about a minute or so to just get all of your materials sorted out before we continue. All right, hopefully everybody is sorted out. Let me know if you need a couple more seconds. But all right, we're gonna go ahead and move forward. So now that we have all of our materials, included our marshmallows, which are separated by color, let's go over what the colors of our marshmallows are. So just like we stated previously, in DNA, we have 4 different types of bases. And those bases are adenine, thymine, cytosine, and guanine. So now we have 4 different color marshmallows that are going to represent those 4 different bases. So our yellow marshmallows are going to be our cytosine molecules, or our C's. Our orange marshmallows are going to be our guanine molecules. Our green marshmallows are going to be our adenine molecules. And our pink marshmallows are going to be our thiamines, or T molecules. One more time. Your orange molecules, or orange marshmallows, are going to be your guanines, or G's. Your yellow marshmallows are going to be your cytosines, or C's. Your green marshmallows are going to be your adenines, or A's. And your pink marshmallows are going to be your thymines, or T's, okay? So now that we have that sorted out, we have to figure out what DNA sequence we're going to use. And your DNA sequence is just simply the series of your bases as they go down in the DNA molecule. So today, I've chosen this sequence, but feel free to choose and use your own sequence. So if you want to follow me, go ahead and write down the sequence, or I'll keep it right here so you can see it the whole time. But we are going to be using the sequence T, A, C, G, T, A, T, G, A, A, okay? So I'm going to keep this here so we can follow it as we build. So how are we going to build a molecule of DNA out of Twizzlers and marshmallows and toothpicks? So first things first, your Twizzlers are going to represent your DNA backbones. So for now, we're just going to work with one Twizzler. Now, you're going to want to choose one of your bases. And now, since we're starting at the top of our sequence, we're going to choose a T or a thymine. And remember, thymines are pink. So go ahead and grab a pink marshmallow. Then you're going to want to grab a toothpick, which is going to be the rungs in your ladder. So again, your Twizzler is your backbone, and your toothpick and your marshmallows are going to be the rungs. You're going to put one pink marshmallow on your toothpick rung, and that's going to represent T. Next thing you're going to want to do is push your rung of your ladder into your Twizzler backbone. So now we have 1 T or purple marshmallow on 1 toothpick, which then has been inserted or pushed in to our Twizzler backbone. Okay, so now that we've basically got the gist of this activity, which is choosing our proper marshmallow color, pushing it onto the toothpick and adding that toothpick into your Twizzler backbone. If you guys have any questions as of now, feel free to send me a message via, again, the chat box, or feel free to unmute yourself and chime in and ask now before we continue. So right now you should just have 1 marshmallow on 1 toothpick, and that toothpick should be inserted into the Twizzler, which is your DNA backbone. All right, we're gonna go ahead and continue. You guys seem to get the gist of it. So, in case you missed it, we have 1 rung of our DNA ladder stuck into the backbone with 1 pink marshmallow representing thiamine or T. Now, we're going to continue this process here of pushing on the next marshmallow onto a toothpick and then pushing it into our DNA backbone. So you guys can work ahead, or you can take your time and follow me as I work. So our next base is A, or adenine. And remember, we said that the green marshmallows were adenine, okay? So again, we're gonna take 1 adenine marshmallow, green. We're gonna take 1 toothpick. We're gonna stick that adenine marshmallow onto our toothpick, and then we are going to insert the toothpick into our Twizzler, which is our DNA backbone. You might have to push a little bit, but once you get it in there, it'll look like this. So now we have 2 rungs in our ladder, and we're going to continue on. So our next base is C, or cytosine. Remember that our yellow marshmallows are our cytosines. I'm going to grab another toothpick, push on my cytosine, and then push this cytosine into 1/2 of our DNA molecule. Next is G, or guanine. And guanines are orange. So go ahead and we could grab an orange marshmallow, stick it onto a toothpick, and then add it to our DNA's backbone. Next up is T, or thiamine. And remember, thiamine was our first one, so it is pink. So we can go ahead and grab another pink marshmallow, place it on our toothpick, and then go ahead and push it into our DNA backbone. Next up is another A, or adenine. So adenines were what color again? Green? Great. So we're going to go ahead and grab a green marshmallow and place it onto our toothpick here, and then place this in our DNA backbone. Now we're at T. Let's grab another pink. And you can take your time and build your DNA molecule. I'm just going to keep moving along, reading my bases and matching them with their color-specific marshmallow. Is everybody good? Everybody moving along nice and fine? Again, if you're not, just let me know. I'm always here. And I'll give you time if you're behind. Again, be really gentle when you're adding your toothpicks or pushing your toothpicks into your Twizzlers. They could be, your toothpicks can break sometimes, or sometimes you could squish your marshmallows. So just take your time and very gently build your first strand of DNA. And my last two here are the same, so I'm just going to grab 2 marshmallows and two toothpicks and get them done in one fell swoop. So now that we've followed our sequence and built our first... So I'm going to pause right here, and I'm going to type out the sequence as somebody has requested, and allow you guys to catch up with building your first strand of DNA as your backbone. So if you need the sequence is now in the message box. So it's TACGTATGAA. No. And again, you guys can go ahead and finish up your first strand of DNA. Remember, DNA is 2-stranded, and those 2 strands come together via the hydrogen bonding through your base pairs. Sorry, I shared that sequence privately. Let me go ahead and share it again with everybody publicly. All right, so everybody should just about have their first strand of DNA sequence completed. So we're going to go ahead and continue on with our lesson. Of our DNA strain by adding the marshmallows that corresponds to our DNA bases, we now have 1 strand of DNA. Well, DNA comes in 2 strands, and its other strand is complementary. So remember, we said that our bases can match up in very specific ways. Those specific ways are T's or thymines always pair with A's or adenines, and G's or guanines always pair with C's or cytosines. So marshmallow-wise, that means your guanines or your orange marshmallows always match up with your cytosines or your yellow marshmallows. And your pink or marshmallows, which are our thiamines or T's, always match up with our green marshmallows, which are our A's or our adenines. So remember, T's always go with A's or thiamines with adenines. And G's always goes with C's or guanines with cytosines. Got it? So now we're going to go back through our DNA molecule and match up our marshmallow base pairs. So if we have T here on our first marshmallow, then T or thymine always matches with A or adenine. So that toothpick also gets a green marshmallow as a base pair. Now we're going to go down our strand of DNA and add all of the proper base pairs. So T's always goes with A's, so I'm going to add a pink marshmallow onto this toothpick as thiamine's base pair. Yellows or cytosines, and cytosines always match with guanines, so C's gets a G or a orange marshmallow. Everybody good with their base pairing? So remember, it's A's and T's and C's and G's always go together. And an orange marshmallow, or good job, Beckus. E for guanine always matches with cytosine, which is our yellow marshmallows. So we can go ahead and add that base pair. And I'm just going to keep working down the line, and you can take your time to work or follow me. So now that we have all of our bases paired, we can go ahead and add our second side of our DNA molecule, which is another Twizzler backbone molecule. So this might take some time and effort, and be gentle. So again, you should have all of your marshmallows now base paired together, meaning you should have 2 marshmallows on 1 toothpick, and remember, it should have its specific buddy. Now, this might take some time and some patience, but basically the next step is going to be pushing on the other DNA backbone or the other Twizzler side onto each piece or each side of each toothpick or each rung in your DNA ladder. So while you guys are pushing in your toothpicks on the side of your DNA ladders, I'm going to take a second and address a question that I also addressed in the message box. So the question asks, why do the bases go in that specific order? Now, DNA bases go in specific orders according to what gene they code and represent. So for this sequence that I used here, I just chose a random sequence of DNA bases and just typed them out and listed them for you. But you could always go online and Google the sequence for, let's say, the protein hemoglobin. So DNA encodes for all the proteins that we need to work in our body. So if you wanted to make a DNA sequence or a Twizzler DNA specific to, let's say, hemoglobin protein or insulin protein, you can go ahead and Google the DNA sequence for those proteins and just choose a few base pairs and build your Twizzler DNA model as such. So again, this is a random sequence that was just made up by me, but if your sequence is specific, those specific sequences then code for specific proteins. Any further questions, just let me know, but hopefully I answered that clearly. We're going to go ahead and continue on with adding our second side of our backbone. That all of our bases are paired up properly. Again, A's with T's and C's with G's is seen here. We could now go ahead and connect our other backbone to complete our DNA molecule. So we're going to do this by pushing the other end of our toothpicks through this Twizzler or backbone piece of our DNA molecule. Now, I'm going to warn you, this takes a little bit of patience, so please take your time and push gently, as you can break your toothpicks or squish your marshmallow nucleotides. So as you see here, I'm just going to hold the toothpick, which is the rungs on our ladder, and also hold our Twizzler firmly and go ahead and push that through. And just move on to the next one and push that in as well. And just keep working down the line until all of your rungs in your ladder are pushed through each side of your DNA backbone. What helps me is to hold the toothpick like this and then to push. So hold the toothpick flat like that and then to push it in as so. Therefore, you don't squish any of your marshmallows and you don't break your toothpick. So just keep working down the line until you connect your DNA molecule together. I see the Beccas have finished. Good job. Twisting into a double helix. So while you're building the rest of your DNA strain, one of your lab mates has asked, how would you alter DNA? You might have to go back and fix any ones that have popped out, and that's fine as well. Just again, be gentle and try not to break your toothpicks. If you break a toothpick, just grab a new toothpick and stick new marshmallow ons and make a new base pair and insert that wherever you need it in your DNA molecule. All right, so we are all put together here. So you can see that, again, our DNA molecule is very simple to the model that we started with in the beginning of this video. So you could see that with the two backbones being red and white and the base pairs going in the middle of our model, we now have two backbones being our Twizzlers, and we have our base pairs matched up properly going down the center as the rungs in our DNA ladder. So now we have our completed Twizzler DNA model. If you want to, you could go ahead and give your model a twist and see that it forms the double helix structure just like real DNA and like our DNA model that we were looking at earlier. So with all these yummy base pairs now made, take 1 second to review which DNA base pairs come together to make those complementary DNA strands. So remember, T's always match up with A's, so thiamines and adenines, and G's always match up with C's, so guanines with cytosines. Go ahead and enjoy your experiment as your afternoon snack, and be sure to share with any siblings or your parents if you have any. Okay, so hopefully everybody has created their DNA molecule. If you had your materials, if not, hopefully I walked you through close enough so that you learned the importance of your DNA backbone and also your base pairs. But again, This can be your afternoon snack only if you've already had lunch. Please brush your teeth afterwards and make sure that your hands are clean while you're eating your snack. But go ahead and pay attention to each one of those marshmallow or candy base pairs as you're enjoying it, and try to remember which ones always pair up. So remember adenines and thymines, or A's and T's, and cytosines and guanines, or G's and C's, always are complementary base pairs because of the hydrogen bonds that they have to share to hold those two sides of the DNA backbone together. So while we were finishing up our model, again, we had another great question. And that question was, how would you alter DNA? So naturally, our DNA can be altered by things such as UV rays from the sun, or any other radiation, or some chemical contaminants that can mix in with our DNA and stop our DNA from being copied properly, or stop our DNA from being decoded properly so we can make our proteins. Now, here in the lab, we can synthesize any piece of DNA basically that we want by telling an instrument which base to add on to the end of a DNA sequence. So again, I could make up my own DNA sequence and build that with a specialized instrument in the lab, or I could, let's say, again, take some UV rays and put my DNA in a UV box that we have here at the lab, and that will actually make cuts in the backbone of DNA. So you won't have your long, nice piece of DNA anymore. Instead, it will be chopped up. And then again, you can't read or decode chopped up DNA to know which types of proteins we can make. So you can naturally alter DNA, and we also do it on purpose here in the lab using specialized instruments. And again, chemicals or UV rays can denature or alter your DNA strands. And what that would lead to is either your DNA not being copied correctly, because now you've changed the sequence, or that DNA could be copied correctly, but when it's decoded to make protein, it's going to make the wrong protein or the incorrect protein, which cannot be used by the cell. So I hope that clears that question up. If I didn't, just again, let me know through the message box. But I'm gonna again keep scrolling and see if I can catch another question here. So another question, can I take a piece of DNA and make it into an X? So essentially, yes and no. So our DNA, again, is going to be in the shape of a ladder. So with your phosphate and sugar backbones, and then with your bases in the middle as the rungs of a ladder. So because this is how two strands of DNA can complementary base pair with each other, it kind of solidifies that it could only form that ladder structure. But then that ladder structure, as you guys could see, can twist. And as you twist it, it kind of condenses or gets smaller and smaller and smaller on itself. Now, once that DNA condenses or gets smaller by twisting in that alpha helix form, then that DNA can form chromosomes, and chromosomes are in the shapes of X's. So within those chromosome X's, we do still have that laddered shaped DNA. So I hope that answered your question. So your chromosomes are in the shape of an X, but within those X-shaped chromosomes, you still have your ladder of double-stranded DNA. Any other questions? All right, so we have 5 more minutes. So before we started, remember we had a poll just to see how much you guys knew about the structure of DNA in general. So now we're going to share that same poll. Now at the end of your lesson, I'm going to give you another 5 minutes, and we're going to go through and try to answer those questions again. But this time, I'm going to share the answers with you. So let's take our last 5 minutes and try to complete our poll. So another question we just received while you guys are polling is, can you alter DNA to make a superhero like Captain America? So, not that I know of in real life as of yet, but one example I can speak of is an experiment where scientists took out the piece of DNA or the gene that codes for jellyfish to glow green. And then they added that DNA into a bacterial cell. And guess what happened? The bacteria then started glowing green as well. So that process in of itself, it's called cloning. So not in humans, but just in simple organisms like bacteria and viruses and parasites, scientists have developed ways to take pieces of DNA from other organisms, such as jellyfish with their coloring, or some fish have like a natural antifreeze, and that gene or piece of DNA that codes for that color or that natural antifreeze that fish and jellyfish can have has then been put into bacteria and or plant cells to give them those same qualities. So I hope that answered that question. All right, we have about 1 more minute on the poll. See if you could use any of the information that you gained throughout this lesson, and I hope you guys had fun. If you had fun, can you send me a smiley face in the message box? And once again, we're going to be doing this every week. I think we're going to try to stick to Tuesdays at 1, but we're going to be doing this every week. So either I will be here hosting or Miss Cynthia from the city location will be in here hosting as well. Oh, thanks for the smileys, guys. Okay, we have about 30 more seconds. And my thumbs up, thank you. Okay, so now we're going to go over the answers. So again, you guys already knew a lot about DNA before we started this lesson. And again, this was just some background introductory information and an activity to the structure of DNA and why the structure of DNA is important. So question number 1, what does DNA stand for? So the majority of you got this one right. And again, DNA stands for deoxyribonucleic acid. So deoxyribonucleic acid, that is what DNA stands for. So number 2, what are the 4 letters that stand for the nucleotide bases in DNA? So that would be the last option, which is A, T, G, and C. And everybody got that right, so good job with that, guys. A, T, G, and C, very important bases. That's what all DNA molecules are made out of. All right, where is DNA found? So DNA is found in all living cells, But in eukaryotes, DNA is found in the nucleus. So that's the name of the organelle where it is. So the nucleus is where DNA is found. Ribosomes are used to make proteins. Right. Number 4, which molecule is not in the backbone of DNA? So remember the backbone or the Twizzler strands? That is made out of a phosphate and that deoxyribose molecule. So only in the backbone is phosphate and deoxyribose. And then the nucleotides are adenine, thymine, cytosine, and guanine are in the middle or the rungs of the ladder. So which one is not in the backbone? That would be guanine. Guanine is not in our DNA backbone. It is one of the bases or the rungs in the ladder. And last but not least, who has DNA? So we have plants, which are made out of cells. Animals, like me and you, are made out of cells. Microorganisms, such as bacteria and viruses, or all of the above? And the answer is, like you guys pointed out to me, it's all of the above. So we know that bacteria and viruses also have DNA. Animals, as us, we have DNA, and also our plants have DNA as well. So I'm really proud of you guys. Again, you guys know you knew a lot of this information before we even started, but very, very good job in completing this activity and lesson. And I want to thank you for joining our first Citizen Science Lab Zoom lab. And again, we will be back next week. So check out the website for registration and make sure you check the materials list to make sure you have all of the materials before the activity starts if you want to participate. And again, you can go ahead and send me any more comments or questions that you have. But if you are done looking at me, which it was great seeing everybody that I can see, you can go ahead and sign out of our meeting. And again, thank you for participating and joining us today. And hopefully I'll see you soon in person. I miss all of you guys that I already know, and I look forward to meeting those new faces that I do not know yet. Oh, hey, River, don't be that sad. Don't cry. But it was great seeing you guys. Thank you so much. See you next week.